everyone. Uh, so part of this talk is going to cover a little bit of work that I did in the postdoc, and later I will be presenting some preliminary data which I generated. And I'm sorry to break your heart, I am not currently working as such in AI and ML. I'm working using some information theory measures or some Granger's causality with my collaborators in a Trivili department. But as of now, I'm not actively looking into AI and ML, which is something which I look forward to discuss with you all in terms of collaborations. So to recap a little bit of brief journey, my training has been in the field of sudden unexpected death in epilepsy. I was mostly trained in physiology, MRI, MEMRI, later followed by auditory neuroscience, where I was working in a hardcore neurophysiology lab. And today part of the data that I will be discussing is from my postdoc work, a little bit of my postdoc work, where we are looking at the top-down cortical projections to the thalamus. What is the role? Why do we need to have top-down projections? What is the role of them? Then followed by which this work, the impact of aging on the basal forebrain projections. Oh, okay. So I hope everyone is able to hear me. Okay. So next part of the work is going to be kind of like which we are working currently on, for which I will just show some preliminary data. We have some proof of concept kind of thing going on, where we are looking into the basal forebrain projections to the auditory cortex. There are different types of projections which you can tease into by using optogenetics. And then third one is looking into the hearing loss, the effect of hearing loss on mental health. There is a huge amount of correlative data that has been showcased in 1990s onwards, but then only it is recently that we started to appreciate the impact of hearing loss on mental health. So apart from these work, we also are working on different other projects like epilepsy or edible electrodes and biomedical devices. So if you want, I'm willing to discuss on those things in terms of collaborations. So going forward to the introduction, the sound waves are nothing but rarefactions and condensations. It's a mechanical properties of the sound waves that kind of like are transduced into electrical signals in this region called as cochlea, that is kind of like a snail-shaped organ, and after once it is transformed into electrical signals by this mechanotransducer, it goes up. It is due to the movement of the stereocilia, wherein the potassium channels open up and leading to the entry of potassium and calcium, leading to depolarization, and so then action potential is generated. Oh, even the point. Yeah, okay. So this action potential is generated, and this action potential travels from cochlea to the cochlear nucleus, lateral lemniscus, superior olivary complex, inferior colliculus, thalamus, and auditory cortex, wherein the sensation of or the perception actually starts for us to perceive the sound stimulus. The auditory objects, one of the famous papers that was published by Yale Cohen from UPenn, have showcased that the auditory objects are formed at the level of auditory cortex, and there, by using the interaction between the auditory cortex and the cingulate cortex and other regions of the brain, is where we actually the perception of the sound starts to happen. And so people who have hearing loss, there is only one way out right now, which is either using hearing aids or cochlear implants. And almost, if we are talking about in India itself, 10 crores of the population, which includes different types of hearing loss, either it could be noise induced or age related hearing loss, that much amount of population are affected. And especially in aging population above the age of 65 years, there is an increase in the, like 1/3 of them are affected by the hearing loss. And the current therapy, there is no therapy, pharmacological therapy, there is not available. There are some gene therapy that are being kind of like proposed successfully in clinical trial 1 and 2 stages. They are there, but apart from that, not much is going on. And currently, the brain-computer interfaces like hearing aids or auditory brainstem implant or cochlear implants are proposed as the next strategy against it. But they have the problem that they cannot hear people in a noisy background, which is called as a cocktail party effect. And there is, in presence of some wind noise, again they cannot hear. And certain frequencies of sound they cannot hear. So here I would like to play a little bit of video which showcases from Arizona State University, where they are doing in a cochlear implant patient. She has one side intact, the other side she is having a cochlear implant. And she is asked, what does she hear from her cochlear implant? So here is such video. Is it going to take more time? It's okay. So I will just do, it's okay, I can just talk about it. Okay. So basically the girl is like, if somebody is saying, hey, how are you? The cochlear implant patient listens like, how are you? Something like that. It doesn't sound exactly, hey, how are you? So that's how they are able to listen. So current technology is lagging behind in that. There is a lot of, basically the current cochlear amplifier or whatever we are talking about, the technology that is being used is they have A to D converter, and then an amplifier, and then D to A converter, which is again converting back it to sound. And there are a lot of theories that have been proposed that suggest that we need to take into account the cognitive factors also to see the plastic changes, the neuroplastic changes that are happening at the level of the auditory cortex and higher structures to account for these differences and to address these issues. So as you could clearly see in this slide, after the mechanotransduction in the green, there are kind of like basically the rest of the auditory structures act like filter banks, and you are kind of like getting your signal. And then once it goes up, the auditory scene analysis, object formation, and the semantic knowledge, all those things are coming up from our cognitive resources and emotional resources. We kind of like form an emotional contact with it, so it is connected with the limbic circuits also in addition to the cognitive circuits. So hearing is an active cognitive process. It's not just a sensory system. That is something which I would like to get through to you. That's the first point of this. And there are certain diseases like aging or noise exposure or infections and sudden loss of any primary sensory input. These things can impact this whole mechanism that is going on, the interaction that is going on between this, and our perception is finally affected. So in response to these kind of aging or noise exposure, what happens is the homeostatic plasticity that is there, that kind of like kicks in, and there is kind of like generation of the sound input or other stimuli that is not there. It's kind of like happens, and that is what we classically see in case of tinnitus, the ringing sound in the ear which people hear, that is not actually there, but we do hear it after hearing loss. It is because of the maladaptive plasticity. And the current hearing aids or the cochlear implants which we have, they are currently doing the digital signal processing, but they are not taking into account the changes that are happening on the cognitive side of the brain, which should be taken into account for them to be able to function in a better manner. So today, now I will start the first part of the talk, which is about like the age-related hearing loss, and almost it affects 6.3 crores of Indian population. Here on the right side, you could see two signals. There are two sound waves. The bottom one is one without gap in between, and the top one is the one with the gap in between. And usually, people who are young, they are able to better detect the gap that is present, the 3 millisecond gap that is present in between the stimulus. Whereas with the elderly, with the increase in the signal to noise ratio, there is the decrease in the signal to noise ratio. In the elderly, you can see the higher thresholds that are present, suggesting that people who are aging, their temporal processing is becoming slower. They are not able to process the information at the same speed as a young person. And then I would like to change the gears a little bit and talk about this protein called as GABA, or GABA neurons that have a protein called as GAD67, which we are using as a protein marker for the GABA neuron expression. And people have shown in studies that in comparison to young animals, in aged animals, there is a decrease in the GABA expression. And this decrease in GABA expression has been shown to be causally linked in causing temporal processing deficits that are seen in aged individuals. And typically in case of like aged individuals, what they do is they use their top-down resources because their temporal processing is not fast enough. They use their top-down resources to kind of compensate for their deficits. Like you might have like grandpa or grandmom or who are elderly people. If you go and talk to them few sentences, they can't hear well. They say that, okay, kya, or what, say it again, or repeat it again. Sometimes they say it, and sometimes they even assume based on the context of whatever you're talking about. So they are using their top-down cortical resources, where in MRI study they have showcased that these cortical regions that are cinguloopercular network are getting activated. So now I would like to do a little bit of exercise before I go into the topic. I want you to point out in the center of the screen there is going to be a stimulus that is going to pop up for 100 milliseconds. Please raise your hand if you can see the stimulus or the red dot. Okay? Now, everyone saw the red dot? Okay, good. Now again, the red dot is going to come up. Please let me know if you are able to see the red dot. How many of you? Okay. Maybe if I repeat it more times, you are finally able to see it. It's not evident on the first time. So that is a topic which we call as salient stimulus, where in the first case, the red was very obvious among all the greens, whereas in the second case, it's like needle in the haystack. We can't see much better. So that is called as a less salient stimulus. So in the following study, we have used salient and less salient stimulus to tease out how our brain works. So we are hypothesizing here that the auditory thalamus is not just a relay of neurons that just pass the information to the cortex. There is a role that these thalamic neurons play in that they receive inputs from multiple structures, including cognitive resources from the auditory cortex, and also sends information to the limbic structures such as amygdala. So in this experiment, this is part of my postdoc work where we have inserted tetrodes, which are all 13 micron wires, a platinum iridium wire that is inserted into the auditory thalamus, and this is a rat that is showcased in the bottom. And on the top, we are seeing single units, or the single neuron that is each action potential, each line that is barcoded, that is an action potential. And in the bottom trace, we are looking at the local field potentials. And here is the animal in which we have injected at the level of auditory cortex, the region which we think is sending in cortical resources to the auditory thalamus. And we see that there is a good amount of co-localization of VGLUT1 and ARCH-T. ARCH-T is the optogenetic expression that which we are inducing to control the neural activity, and VGLUT1 is a cortical neuron, pyramidal neuron marker. And so here is the circuit. From the IC, the excitatory and inhibitory projections are coming on to the MGB, from where it is going to the auditory cortex, and auditory cortex receives the inputs back to the MGB. And we are going to cancel out these inputs that are there. And So here we have two different types of stimulus, salient stimulus, the first one, and the next one is the non-salient stimulus. All of them are non-salient stimulus, similar to what we showcased before. And so here is one single neuron from which each dot represents an action potential. And you could see the stimulus were presented either in a random way or in a predictable manner. And we see predictable manner means the frequency 2 Hz is repeated 10 times, followed by frequency 4 Hz for 10 times, which is there on the y-axis. And this is a 500 millisecond snippet on the x-axis. And we see that there is a kind of like almost similar response in both random or predictable presentation. The neuron is almost responding similarly. And then we increase or decrease the salience. And what we see is that the predictable responses, there are more neurons that are firing. And then we are optogenetically inhibiting both during the random presentation and predictable presentation. And what we see is a huge inhibition at higher frequencies, which are more challenging for us to follow when we are blocking the top-down resources from the auditory cortex. We see a reduction in the firing rates. And then followed by which, once we stop this shining of this laser light, we see the recovery in this single unit. So the take-home message here is that when the stimulus is salient or 100% depth, there is balance between the bottom-up and top-down information. But once you tilt it to non-salient, then there is increased usage of cortical resources in order for us to understand what is the stimulus. And when you block it, the top-down effect is gone. Now, this is a project which I have started in our lab, wherein we are looking at the differences between a young brain and aged brain in terms of the basal forebrain projections. In age-related hearing loss, which impacts almost 10 crores of Indians, this hearing loss leads to maladaptive neuroplasticity at the brain, wherein in young animal, most of the research currently has focused on cholinergic neurons in terms of like cholinergic neurons increase the salience, are more kind of like promoting the cognitive activity and all that, but not much is known about the other neurons that are present in the basal forebrain. In fact, cholinergic neurons only are 20% of the whole basal forebrain population. So what happens to all these neurons? What is their function? That we don't know. So this part of the project investigates functionally how it impacts the auditory cortex and what happens with aging. So that is something which we are currently looking into. This is the electrode setup which we have. The mice is anesthetized and we are having a thin tungsten electrode which is recording the electrophysiological activity from single units. And here we are seeing the spikes that are kind of like face locked to the auditory stimulus. So the top trace that you're seeing are the action potentials along with the local field potentials, and the bottom colored traces are the ones which are showcasing the actual sound input. So there is a huge face locking which you can see at the level of auditory cortex. Right now we are performing the experiment still, so I don't have much data in terms of that. And in this same project we are looking into what is it that the rats can hear. In case of like humans, we can ask like what frequency they hear, what sound they hear. That is called as pure tone audiometry in an audiology clinic, but unfortunately the rats can't use they don't understand English. So, okay, so here is an operant conditioning behavior wherein the stimulus is presented and the rat is trained on like to go and get its pellet or the food that is available. So this is following the Pavlovian or associative learning that is happening, and through which we can understand when the light is on, it goes, and for a particular stimulus, it is trained to pick up its food pellet, which is its reward. Basically, they are kept on some diet. So the motivation is that they have to go and get their food, so they have to work hard. And so we will be understanding how it is functioning. So as of now, I don't have much data to show about this project, but it's just that we are adding another variable here, which is basal forebrain, and how the basal forebrain inputs are impacting this whole network. Now I want to switch gears to noise-induced hearing loss. We are exposed often to loud noises. Probably in this talk, we are having some 80 to 90 decibels. Those who are sitting in the front row, caution. And those who are kind of like attending DJ parties and other things, or having a stop at a traffic signal, you are getting probably exposed to 90 dB, 100 dB in aeroplane, 100 dB. And so as we talked about, hearing is an kind of like a cross-modal activity wherein the auditory network interacts with the limbic as well as the cognitive networks for us to perceive the sound stimulus and understand what the opposite person is saying. And so here you could see in the Lancet report, which is showcasing an increase in the number of cases of noise-induced hearing loss that affects almost 10% of the population. And studies in JAMA and Lancet have reported that most of these people have mental health issues like anxiety and depression, and also cognitive issues such as dementia. This study, my student here will be presenting it as a poster. I'm not going to go into the data as such. I will be just talking about the ideology or the rationale behind this whole thing. So studies have showcased that there is, in people who are working in military, there is a working memory impairment who are exposed to constant noise. And also similar studies have showed up that there is a decrease in the functional connectivity between the brain regions such as amygdala, auditory cortex, and anterior cingulate cortices in noise-exposed patients, which you can see onto the graph that is showcased. And that people may say that this is correlative study, but there is also evidence, a recent Irish study that is published in 2023, where they have showcased that once you restore the hearing with hearing aid, the opposite is happening. So here they have evaluated pre and post the mini mental state exam and Montreal cognitive assessment, wherein basically you are asked like tasks like what are the objects that you can see, can you reproduce it, and they are asked to kind of like repeat the words, what day is that today, all those kind of like how is the weather today, simple questions. And people do answer that, and pre and post these assessments were done before the hearing like pre-hearing aid and post-hearing aid, and people have found that there is a huge recovery that is happening in patients who used hearing aids in comparison to those who didn't use hearing aid. And some preclinical findings also have showcased that in the first chart you could see the auditory cortex in control situation and noise exposed animal. There is more increase in the activity, which is shown by more yellow color, whereas in the prefrontal cortex, usually which is connected from auditory cortex to A1, the information goes, and usually you see activity in the prefrontal, but after noise-induced hearing loss, that activity is gone. So there is a deficiency in this functional network. And structurally also many MRI images have shown atrophy and other things happening, and we are just discussing the functional studies here. And another study which was done using patch clamp in control animals, they have showcased that the FEPSPs that they are measuring from the dentate gyrus cells after inducing the high frequency stimulus mediated long-term potentiation in control animals, the black, they are pre-condition, and post high frequency stimulation is what that is shown in green color, and you could see an increase in the synaptic long-term potentiation, whereas that thing is absent in noise-induced hearing loss animals, suggesting that the functional connectivity is lost, the neural plasticity is lost in these animals. And here is a brief gap that depicts all the increases and decreases that could be the aftermath of neural degeneration that is happening because of excitotoxicity. And again here we find that there is a deficit in the GABA, and once you could see the paravalvular neurons that are a specific type of GABAergic cells, again we see a significant loss in that. And again, in preclinical findings, we see that the neural regeneration is not happening, and high expression of p-tau also is happening in patients who are having hearing loss. And so the take-home message here is that with the hearing loss, what happens is the MGB to auditory cortex connections are not so functional. The increase in excitation happens with the decrease in inhibition. which leads to a lot of increase in the excitation or excitotoxicity at these structures such as hippocampus, prefrontal cortex, anterior cingulate, and amygdala regions, and which could impact the behaviors that are usually controlled by these brain regions. So in conclusion, the part of my postdoc work was on auditory cortex and how it impacts the auditory thalamus. And then later, I showed you some hypothesis on how basal forebrain could affect the auditory cortex and how we don't know much about different neurons in the basal forebrain, what is the impact of them on auditory cortex. And then we talked about how auditory cortex that is connected to different limbic and cognitive structures, how it can impact it. And then finally, this is just a small picture. The bigger picture is this. We have even more networking among all these brain regions.
Long live India and France friendship.